Welcome back. Please have a seat. So welcome to the last session of today. So we have 1 hour for a couple of presentations. And the first presentation will be by Luis on the role of capacity building and updated FEO guidelines to support the banking strategy. So Luis, please.
Okay. Thank you, Sipke. I will go briefly through the training program that we have followed in IMAGE in the framework of Work Package 7. I've listed here the names of several people who were involved in the training activities. Sorry if I missed anyone. So the objectives of Work Package 7 were to demonstrate the benefits provided by animal gene banks and promote their visibility and transparency, disseminate new knowledge, and strengthen human resources responsible for managing animal genetic resources conservation programs, both in Europe and in third countries. And this is the focus of my talk today, is mostly about the training part. Now, these capacity building programs were designed to strengthen the activity of gene bank managers and other stakeholders involved in managing animal genetic resources in Europe and third countries, as I mentioned, but we had to have in mind that these are very diverse groups and needs in terms of the role that they play. These were either gene bank managers, PhD students, research, and so on. The scientific background that they had, from very highly skilled people to practical technicians, and the geographical location, which also placed the language barrier because the courses had to be in the language that was most comfortable for the people attending the courses. Overall, let me go from what we had planned to what we achieved. Initially, we had planned to have 3 courses in Europe, 2 courses in third countries. By the end, we realized 3 courses in Europe and 5 courses in third countries. So actually, we were able to accomplish more than was planned from the beginning. Just to give you some idea of the various courses that we had in different places, these are some pictures that I'll show later on. The first course took place in Argentina in March 2018. This is in Spanish, but that was the poster that they used, was of course in Spanish because the attendants were Spanish. But I suppose you can understand what the title was. This is the group of people who attended the course. We had 22 participants in situ, 16 from Argentina, from various parts of Argentina, 3 from Peru, 2 from Uruguay, and 1 from Bolivia. They all came to attend the course. And the course took place in Balcarce, which is a small town located here south of Buenos Aires. And the local organizers were INTA, a partner in IMAGE, through Maria Rosa Lanori, who is here somewhere, right there, and Lilia Melushi, who is not here. We had 3 lectures from different IMAGE groups, and all the lectures were in Spanish. In addition, we had also the possibility of providing Skype, talks to people in other places. So we had a group of people, 15 people in different places in Colombia who took the course through Skype. It worked relatively well, I think, the Skype part. In addition, we had a field trip to see Creole cattle in the region, and the stakeholders meeting with participants, mostly Creole cattle breeders, 32 participants in the stakeholders meeting. Now, the second course was in Colombia in October of 2018 on conservation and management of animal genetic resources. And this is the poster that they developed for the course. So now the attendants were in place. We had 33 from AgroSavia, which is a partner in this project, and the universities and so on, 33, and then we had 7 students attending the classes by Skype in Argentina. We had 3+1 lecturers from IMAGE. One was Paul Butcher by Skype. He wasn't physically there. And these were the 3 participants, and all the classes again were in Spanish. This is the group of participants. The course took place in Bogota, the capital, and then we had a field trip then the last day to Villavicencio, where they have native pigs and Creole pigs and cattle and so on. Now, this is a brief outline of the course. I'm not going to go through the whole list, just you can see the topics are basically the topics of how you manage, how do you characterize and how you manage genetic diversity in the framework of conservation programs. And the course ended, like the one in Argentina, this one also ended with presentations by participants. I'll give you a brief overview of what happened in this presentation, because I think it was extremely useful, this exercise. Now, the first course in Europe was immediately after this one in Colombia, and it was in Wageningen. The first interactive postgraduate course on characterization, management, and exploitation of genomic diversity of animals, October 2018, 28 participants. The picture is a little bit on the dark side, but that's what we have, so we don't really recognize many faces, but we assume that we know who they are. These are the participants from different countries, several countries represented, and this is the list of lecturers who were involved in the course in Wageningen, the first course in Wageningen. And this course received the education prize of the Wageningen Graduate School of Animal Sciences. It was considered the best course as far as I can understand. So they deserve to be congratulated on that. Okay, this is the outline of the course, the topics covered in the course. So you recognize, I mean, the different topics are all related with characterization and conservation of animal genetic resources. And then in 2019, we had the course in France, organized by AgroParisTech in November. And it was a training session for population managers and users of gene banks. And it was organized, as far as I understand, in the framework of the 3rd International Seminar of CRB Anim. C'est ça? No? It was independent. Okay, sorry. Okay, so the trainees were 15 researchers, of which 11 came from different countries. I'm not going to list them. I'll show you a map at the end. And we have a very nice coverage of the world with the different countries included. The trainers were from AgroParisTech, Institut du Port, Institut de l'Élevage, and Göttingen University. The course outline, you can see here the 3 major topics of the course. This course was, of course, shorter than the other courses. The other one were mostly weekly courses, weekly courses, and this one was for 2 days only. Then this course was quite successful, so they decided to have a second course by the end of last year, in December of 2019, in Wageningen again, on characterization, management, exploitation of genomic diversity of animals. The picture is quite a bit better than the other one. We can at least recognize the faces. trainees 27 from different countries, very diverse group, and the trainers were essentially the same as on the first course. The program outline is, I think, very close to the first one. And then we also had a course in Egypt by the end of 2019, in November, on management of livestock and poultry genetic resources within the scope of climate change. It was organized by the Animal Health Research Institute in Egypt, and the course was, as far as I understand, was in English and some classes in Arabic also. This is the group of students that attended the course, and these are some of the classes. You see our moderator here teaching a class. In Egypt, there were 25 trainees from Egypt, and they were mostly professionals and PhD students. And the course outline you can see here listed. I mean, it's mostly, again, characterization and conservation of animal genetic resources. And then, as well as in Wageningen, the first course was successful, so they decided to have a second one. Also in Colombia, the first course was quite successful, so they decided also to organize a second one. This time, there were 40 students. Most of them were different from the first course, but some were common to the first course. And this was also provided by live streaming to Cuba, Argentina, and Spain. And it worked quite well. The live streaming system worked quite well. The name of the course was Genomics and its Application in Conservation and Characterization of Zoogenetic Resources. The trainers were from IMAGE, University of Lisbon, University Complutense de Madrid, and INRA. It changed the name already at this time. It was January, so it should be INRA. Sorry. Didn't update that. The second course, the outline is a bit different from the first one. These are taken from the students' presentation. So they used either their own data or publicly available data sets, so they used that for analysis and for their own presentations. And this was, again, a very interesting experience. Probably you cannot see the titles of the presentations, but there were some very interesting presentations by the students. All of them presented very nice work, and actually we are trying to organize a session within the Congress of the Latin American Animal Production Association to have them presenting their work in this session of the Michel Nav is trying to lobby for that. So let's hope he will accomplish that. Last course was in Morocco, just a couple of weeks ago. Was in January 2020. Biobanking for sustainable management of farm animal genetic resources, novel approaches in genomics, reproductive technology, and economics. And also there was a stakeholders meeting about cryopreservation of animal genetic resources. The languages of the course here are listed in English and French, but I think also some classes were in Arabic. I understood. No? Just French and English. Okay, sorry. Okay, so this is the group of trainees in Morocco, 20 from Morocco, Tunisia, Nigeria, and Sudan, and the trainers were from INRA Maroc, INRA France. This one is updated, okay. Wageningen and the University of Edinburgh. This is the outline of the course with some differences, but it's basically the same framework for all the courses. And so what have we accomplished? Just to summarize, how far were we able to go? I think that the first thing that we noticed was that there is a major demand for training in these areas. I mean, people really need and show the need for the training in these areas. First one. And I think that IMAGE was able to successfully organize various training activities. Usually these were one-week courses with high quality standards. and we made an effort to be in a language best fit to each group. Involved lecturers and students from several IMAGE partners, and aimed to build capacity in conservation and management of animal genetic resources using novel technologies. And I think we did quite a nice job in this area. So to summarize, we were able to develop courses in eight courses altogether in 6 different countries, 3 continents, Africa, Europe, and South America. We trained nearly 240 students in these courses, representing 36 different countries. So if we draw a map of where the students came from, someone told me this looks like the coronavirus expansion map, but it's not that bad, really. So we really, I mean, some countries were represented only by 1 student, China, for example, but we covered a large part of the globe was covered by these courses. So I think we can be proud of what we accomplished. Now, I'm going to take 1 minute just to summarize the activities of one of our partners, EFAB, on technology transfer to stakeholders. And their activities have been mostly in developing brochures and newsletters to be used for passing the message to the general public. So we had the brochure, 3 newsletters. We still have 1 newsletter to come. Correct, Duru? Yes.
Yes, this is actually since March 2019. We actually
Okay, so it's more. Yeah, you're right. Sorry, it's for a shorter period of time. Yeah. So we have more newsletters than all it is here. Program flyers for EAP 2019. Image demonstration desk in the meeting in the FAB meeting in Dublin 2019. Policy brief and an interview with our boss, which is available on the web page. If you have not seen, you can go there and see. And you have here the contacts of the 2 people who have been mostly involved in Image, Shala and Duru. So if you need anything, you can contact them. Now, I'll give the floor to, sorry, I'm supposed to give this message, remind you that you don't want to miss this meeting in Porto, Portugal, in September, first week of September. It's a very nice place. I'm sorry that Andrea Rossati is not here. He would be glad to see this, but anyway. So I'm supposed to give this reminder to attend the meeting. Okay? And now I'll pass the floor to our colleagues from NordGen, who will give you a brief update on the guidelines and what we will have to accomplish in the next 3 weeks.
3
right?
weeks.
Isn't it?
Next week.
Next week.
1 week. Okay. Okay, thank you, Luis. Just a few words about the guidelines. I think most of you that are here were here yesterday as well. Maybe this is the final deliverable from the project. We promised to deliver some guidelines as a part of Work Package 7. Just to tell you how we went about this. As it said in the title of Luis' presentation, it's basically to update the existing FAO manual on cryopreservation. So this is the one that we have used as a basis for this work. But in Image, we also conducted a gap analysis, a quite thorough gap analysis covering lots of details. We did a brainstorm as well in Work Package 2, so that is the other part of the basis for this work. And the third part is that we have interviewed all the work package leaders about the deliverables, the results from their work packages, how they were seeing them fit into the actual guidelines. And as we agreed before, when we started this work, we basically asked 2 questions. What is new, and what should be revised that is already in the FAO manual? So this is basically what it looks like. On the left side is the FAO manual, the outline of how that is constructed. It has 12 sections covering basically everything to do with cryoconservation and a little bit more. On the right side, you can see the gap analysis as we did it in the Image project. Different headings, but basically we covered the same ground. So now it's up to us in the guidelines to gather all this in a meaningful set of recommendations. I will show you one example, because this is very much a work in progress. This is only started in autumn last year, but only really started here in January. An example from section 5, we suggest a new topic that's called quality management systems. We briefly mention what we actually did in work package 2, what is the basis of any recommendations that we're going to make. I just listed a few bullets, what came out of the surveys that we did. Based on this and based on any other experience we have from this work package, we will then formulate some guidelines. I have given some examples here that what the guidelines could include, and then at the bottom you can see the gap analysis results, just to see if we cover everything that we already pointed at in the gap analysis. So in this way, and this is an outline, we will try to construct the guidelines as suggestions for an update. As we also talked about yesterday, there are parts of the FAO manual that need revision or update that we did not work on in the Image project. Those we will not cover in detail. We will just suggest to the FAO that this and this section needs an update or a revision. So in our own guidelines, we will focus on the work that we did ourselves. Now, as you saw that the FAO manual has 12 sections, I will not take you through all 12 of them. Those of you who were yesterday know why. It takes a lot of time, but the only thing I want to say about this is that we have approximately a week and a half to gather all your input and to come up with a draft of guidelines that is a little bit more than bullets. So those of you that have to contribute, be prepared. Thank you.
Thank you. So thank you, Louise and Jaap. With time for one question. It was a nice overview of all the activities in terms of capacity building, training, dissemination. If no questions, thanks again. Continue. So our next speaker is Manuel Luque from Spain. As you know, FAB, the European Forum of Animal Breeders, is a consortium member of Image, and we thought it would be nice to have a member of FAB, in this case FEAGAS, and Manuel is the director of the Federation of Breed Associations in Spain here, and member of FAB, and we asked Manuel to give some perspectives from the breeding industry on the results of Image or on the Image project. So Manuel,
okay. Good afternoon, everybody. Thank you very much for the introduction. First of all, I would like to express my gratitude for the invitation for taking part of this important event on animal genetic management at European level, and I would also like to congratulate all the Image members project for this important project and for this or for organize this meeting. Well, as you can see in the title of my presentation, I'm going to give you the sector's opinion about ex situ conservation, apart from some extra and interesting information about the situation of the ex situ conservation in our country. Well, this is the content that I am going to cover in my presentation. Firstly, I would kindly like to take a minute to talk to you about our organization, FEAGAS. And secondly, I would also like to give you some information about ex situ conservation in Spain. And to finish my presentation, I want to give you the sector's opinion regarding this important topic that we are discussing here today. Well, let's start with the first point. What is FEAGAS? FEAGAS, this word is the acronym in Spanish of Spanish Federation of Livestock Purebred Associations. It was founded in 1982 as a technical and representative national forum for member organizations. It is currently the largest umbrella organization regarding the livestock purebred in Spain, and FEAGAS nowadays groups 113 Spanish purebred livestock organizations belonging to the different species with interest in animal production in our country: cattle, sheep, goats, horses, etc., all of them officially recognized by the Ministry of Agriculture or the different Department of Agriculture of the different regions in which Spain is divided, all of them to manage and coordinate actions related to the herd books and with the development of the different breeding programs in Spain of the different breeds under their control. Apart from many others, our main functions and responsibility are the preservation, improvement and sustainable use of the livestock breeds in Spain. We also represent, manage and defend the interests of its member organization in Spain and abroad. And we also collaborate with public bodies, institutions, university, research centers and so on in all actions of interest related with conservation, breeding, management and sustainable use of the animal genetic resources in Spain. This is basically our organizational structure. On the one hand, we have the typical or the normal governing bodies of this kind of organizations, and on the other hand, we have different technical working groups, but I would like to highlight these two. One technical committee focused on the local breed at risk with different representatives for each species, and other technical committee for the bigger breeds. I mean those breeds, those local breeds not at risk, that we call in Spain the promoted breeds, and also for the transboundary or the exotic breeds officially recognized here in Spain by the Ministry of Agriculture, and with all of them we have a breeding program in place. This is the distribution of the percentage of the different breeds within FEAGAS. As you can see, the highest percentage belong to the sheep breed, and the second higher percentage belong to the cattle species. Well, after this short presentation about ourselves, I would like to go to the second point of my presentation and give you some information regarding the ex situ conservation in our country. Let's start with the historical overview of the gene bank development in our country, and in this case I would like to highlight that originally we have paid a lot of attention to all actions related to the in situ conservation, but however after some years breeders associations have paid a lot of attention to the breed characterization because of all FAO recommendations some years ago, and also they have paid a lot of information not only to in situ conservation but also to ex situ conservation as well over the last decades. All this has been the origin of the different gene banks for the different local breeds in Spain, which are located in 3 different locations. We have the CENSIRAS, which is the acronym in Spanish of Animal Breeding and Reproduction Centers, the research institutions and the universities. All these gene banks have been coordinated by the Ministry of Agriculture with a very clear objective to try to coordinate all this information regarding all these gene banks for different breeds around the country, and mainly with a very clear goal, trying to set up and strengthen the national gene bank in order to store a copy of the different breeds copied in Spain. This is an institutional organization of the animal genetic resources in our country. We have 2 levels. We have the regional governments. In Spain we have 17 regional governments. These are in charge of the regulation of the smaller breeds. I mean those breeds which are mainly in one region of the country, and on the other hand we have the central government, in this case the Minister of Agriculture, which is in charge of the regulation of the different breeds located in more than one region in our country. These are the objectives of the national policies of cryoconservation. We manage everything based on this national program for the conservation, improvement and promotion of the livestock breeds. This was born from a royal decree in 2008, but based in all the recommendation from FAO after launching and the implementation of the global plan of action, and after that royal decree was updated last year because of the new European animal production regulation. This is the basis to set up our breed conservation programs, and to know very well what kind of tissues it is necessary to store, the minimum of doses, number of donors, and how to proceed with a copy of the National Gene Bank. On the other hand, we have the how to manage or how to proceed with the task group on cryoconservation and how coordinate all these actions, how to analyze it, and what is the criteria to create and manage all these bank coordinated different stakeholders, as we can see here in this slide. In this slide we can see the involvement of different stakeholders. It started from the breeders associations with a clear idea from them to conserve a breed from these main ideas that I explained earlier, in trying to characterize in a very deep way they breed, as FAO recommended years ago, and putting in place an in situ and also ex situ activities. In that case, we have on the one hand the administration in order to carry out the verification and approval of the breed conservation programs, in order to provide facilities for the gene banks, in this case for the animal breeding and reproductive centers, and also providing subsidies or financial support for the conservation of the genetic material to the breeders' associations, and also to verify the implementation of the breed conservation program through the breed inspector. On the other hand, we have the experts in animal genetics, which help and support all this process to the breeders' associations, and all this information and genetic material go as well to the National Gene Bank in order to store a backup or a copy of the different gene banks that we have all over the country for the different breeds. Regarding the legal aspects, each gene bank establishes its own rules to regulate the ownership and realize the gene plasm. All this, as I explained before, go to the National Gene Bank, which is located in a village here in Madrid, close to here, which is where is the location where the genetic material is stored, owned by the breeders' associations. The stored genetic material can only be used for breed destruction, introduction of genetic variability into already established populations, recovery of the gene bank of the breeders' associations in case of destruction, or the release of germ plasm has to be approved by the National Advisory Committee. The results of all this management and working with information and working with genetic materials. According to the information we get from the Domestic Animal Diversity Information System from FAO, we can compare the situation of Spain compared to the average situation of the rest of the countries in the Europe and the Caucasus region. If we take into account in the left side of the screen, we can see the information taking into account the information of all species. We can see here the amount of conservation material, if we have sufficient or we have enough genetic material stored to finish the gene bank for one breed. Is the green color, orange color. We have started to work on it, but without enough genetic material. In red, with no material. In red, with no information. We can see what is the situation, taking into account all species in the left side, and if we take into account only the main species with interest in animal production in our country, cattle, sheep, goat, horses, and pigs, we can see the right side of the screen, and we can see how we have started to work and to store genetic material for more than or over the 50% of the breed of these species in our country compared to the rest of Europe. Here we can provide you with more details going into different species. In this case is the case of the cattle species, and we can see that we have finished or we have completed almost the 60% of the gene banks of all the cattle breeds in our country, and we have started to work with over the 40% of them. And I provide you with more extra information regarding the number of embryos, male donors, semen samples, and so on. Regarding the situation of the sheep species, we can see that we have started to work with the 60% of the breed, and we have finished almost the gene banks of the 30% of our breeds. The situation is a little bit worse with the goat species, but anyway, we have finished almost the 20% of the Spanish goat breeds, and we have started to work with the rest of them, which are going on with the storage of the different material or genetic material of these breeds. This is the situation of the pig species, and this is the current doses at the National Gene Bank, doing a comparison of 2018 with 2019, and you can see how the trend is very positive in all species, less in the case of the horse species, where the situation is the same in these both years analyzed. Well, this is the last part of my presentation. What is the sector's opinion regarding the ex situ conservation after seeing all this analysis of the situation in our country? Well, first I'm going to give you the opinion or the ideas of the local breeders associations. Associations believe that it is essential to have a complete gene bank for the breeders. The initiative to carry out the gene banks arising from them are the breeders, following the recommendations of the FAO and administrations. They also tell us that most of the association think that it is very important to have a backup or security copy of the national gene bank, particularly if they are going to face any big problem in the future with the breeds. The ones that haven't got sufficient copy think that they will get in a short or mid-term. It depends of the resources and the problems that they are facing, but this is what they have in mind. The problems that they face, the main problems are the financial support. They receive financial support from the public administration in Spain to do that anyway, because of the huge number of breeds that we have in Spain, and because the situation of the different breeds is very different in Spain, because most of the local breeds in Spain are in danger of extinction, and many of them have not a lot of resources. They obviously ask more financial support to trying to go more quickly with all this process. In other case, they point out the lack of agreement, because they also explain us that they face some problems when the breeders associations have different criteria than the university which are working with the breeds, or maybe with the local administration from which they need to have any support, and so on. They also point out the animal management, because most of these breeds are very rustic, and they also highlight that they face some problems in order to take all this genetic material, because all of the behavior of most of these breeds, and they also highlight the problems to get the samples fertility, for example, in the donkey species. The opinion of the transboundary breeds officially recognized here in Spain is that they don't think it is essential to set up a gene bank for the breeds, particularly in the case of the global breeds, apart from many other things, because of the quick genetic evolution of this kind of breeds. And they agree that the setting up of the gene bank for the breeds is planned because of the recommendation of the administration, and not because because they have a real interest on it. Like conclusions of these three parts, I could say Spain is one of the countries with the highest biodiversity. Over the 80% of the breeds recognized in Spain are local breeds. The breeder associations of local breeds or autochthonous breeds promote the storage of genetic material for their breeds. The Spanish sector believes in the importance of the ex situ conservation, and over the 50% of the Spanish cattle breeds have sufficient genetic material stored, and the rest it's in process to be completed by breeders associations. And with this idea, I finished my presentation. Thank you very much for your attention.
Thank you very much for the very interesting presentation and for presenting the views of the breeding sector here in Spain. I can imagine there are some questions from Spain.
Yes. Thank you for the presentation. Most of the breed societies in Spain believe in the importance of having the conservation ex situ and the gene banks. Which are the main problems that the breed societies find to complete the gene banks, besides the money? Because of course, I know that one of the most important problems is to get money to develop these actions. But do you find other type of problems to look for a solution to complete the gene banks?
Maybe this is not a problem of a direct finance, but you have to take into account that most of the local breeds are breeds at risk, and their resources are very limited, and then there are many other priorities. They think that this is important, and they want to continue providing genetic material in order to have this goal in their mind. But you know that the situation with this breed, they have maybe in some cases 1 or 2 technicians to do everything, and they want to do that, but they are facing maybe many structural problems, I think. Not in direct financial problems, but indirect financial problems.
Lack of resources.
Yes, yes.
Alessandra.
Now, of course, my question is about the possibility and the willingness of submitting data, information about samples, and in that case, who's in charge, basically, and who should we contact to centralize the effort?
Okay, thank you.
Good to say that also in the room is Fernando Tejerina. Fernando is chairing the network of gene banks actually in Europe, the working group of ERFP. I think you're also in charge for coordinating from a ministry point of view, the gene banks here, right?
The problem with information is that this information is in the gene bank. The gene banks are in the region, and obviously they are the competence in order to send the information. We have a database that is called ARCA, and in ARCA we collect information from the gene banks, but it's not individual information. It's information as a whole. It's the same information as we did as the gene bank sent to Eugenia. Obviously, I think, in my opinion, we have to work in two steps, two phases. The first is collect information as a whole of the gene bank, information of the gene parameters, the number of semen doses, the number of males, and then it depends on its gene bank. Its gene bank then can send more information in detail to the max. But I think a good option for the future, we are working also in the working group in the development of a new software for gene banks. Try to integrate the tool in this new software for gene banks to get the possibility to the gene banks to send automatically the information to the European databases, Eugenia and other ones, with the same management system.
Okay, I'm afraid we have to move on. Thank you again for making time. You are a very busy person, so thank you very much. You are very
Thank you.
to give the presentation. The next presentation is Elisabeth Blesbois. A presentation about the public-private partnership project on protein markers in goat semen. So Elisabeth, the floor is yours.
So the presentation. Okay. So this is just to give you an example of how in image we could at a moment find subjects that have interested, of course, scientists, me for example, but also a private company and a technical institute in France. So, Okay. So in the course, in the work package 3, we developed an approach, a proteomic approach, to make what we call a phenotyping of the semen, to have an idea of the identity of the semen through the composition of protein of the sperm cells. And this tool has been successfully developed in the SHICAM, since in the SHICAM you can have, not so easily, but you can have an access to fertility in very standardized conditions, and also we can get quite a lot of animals. But at the moment we thought that it could be very interesting to develop the same approach in one mammalian species of interest. And so we looked for which company could have some elements to do that. And we knew that a company named Capgen, in France, involved in the genetic of goat species, could have such information. So the information that we needed were to know the semen, to have semen samples, and to know which progeny have been produced with such a semen. And so after, with this tool, we can study the semen and see the profile of protein, and after that, see if there is a relationship between the protein profile and the fertility, or better, the progeny obtained with the same semen. So Capgen was okay also to do that, but we had to manage that, and we had a partner of IMAGE, IDEL, represented here by Coralie, for example. That is a technical institute involved in the relationship between the scientists and also the different companies. And so it seems that this project has been interesting for the three partners, IDEL, that gave a manager, could pay a manager for this project, Capgen, that so the private companies that accepted to give the information on the semen of the males of Capgen and the fertility results, and so we could start this collaboration this year. It's in the last year of IMAGE. Well, it was in 2019. So in 6 months ago we started. Okay, so you have all that. And so the aim is the identification of new quality fertility and fertility markers of goat semen in order to better predict the fertilizing ability of the semen collection stored in the cryobank. This is the objective inside IMAGE. But you understand that this aim may be applied also to other application in breeding. So the three partners involved are Capgen, IDEL, and INRAE, and we based our work on the proteomic tools that have been developed in IMAGE for chicken semen. Okay, so what about the animals that we could get for that? Oh, okay, not only the animals, human first. First, we needed a manager, and so IDEL provided a manager of the project, a methodology of work that was provided by INRAE mainly, but not only, in collaboration with the two other partners, and the biological material that was provided by Capgen. And so the biological material, it was cryopreserved goat semen, or bug semen, from two breeds, the Alpine and the Saanen, so two breeds in France, okay? And Coralie is a specialist of these breeds, we already had some information from the semen because we got the viability parameters of the semen, the motility parameters also, and more important, we know, yes, the number of progeny obtained per ejaculate. Here, the number of progeny, it is not fertility, but in the first approximation, it is used here as a parameter that is not so far from fertility. After that in the future, probably we will refine the system and work on the echographic images during the gestation. So, which I would say, tools, proteomic tools. So 2 different tools we use every time. On the left, that we call intact cell Malditov mass spectrometry. That is a quite simple method where you put a cell, here a sperm, or many sperm, on a specific plaque. And after a process, you can get different peaks that show you the peptides and the protein that are in the range between 1,000 and 20,000 Daltons. So in this area, you have small proteins and you have also peptides, original peptides, but also peptides of degradation. So it's a good place to have an overview of protein and metabolic aspects for that. And the other method is a more classic method of proteomic, but also more difficult to get. That is what we call a bottom-up proteomic, because we start by, well, okay, I don't go inside the technique. So you know 2 different methods. And with that, we make 2 different systems of profiling of the semen. And after that, we make the relationship with what we call the fertility. And in a future action, we will use a modeling for all these tools to get a prediction. But we are not yet at this stage. Just what we've seen first, that there were a very high variability between ejaculates. Okay, so at first we had decided to go on a separation between male, but we see we have first obliged to see a separation between ejaculate, even of same male. We selected the good and bad ejaculates, different, and we made the same work on the two breeds, and then we made the proteomic analysis. So what about the first results for what we call intact cell Malditov mass spectrometry? You see that the alpine, with the alpine breed, you have 191 peaks that were obtained, with the cyanine approximately the same number, and we see that the 2 profiles are not so different, because there are 86% of proximity of the profile between the 2 breeds. This is very interesting, because in the future we will probably be able to join these 2 breeds in order to make a same modeling. Okay, and at this moment, for this method, we have peaks, and now we will go ahead with the identification of all these peaks and with their differences regarding the fertility. Already, we could identify 23 of them that are differentially expressed depending on the fertility. So it's interesting. For the second method, we selected, because it's much more difficult and much more expensive, we selected only 5 good and 5 bad ejaculates, and we made the work, you know, after electrophoresis, etc., etc. And so with this tool, we identify more than 60 proteins, and with 19 of them that are differentially expressed depending on the fertility. So again, now we have another work to do in order to really identify them. So already we know that some of them are already fertility markers known in bovine, for example, or in human. And we know also that some others are not already found as fertility markers. So there is an original part for that. So in the future, we will go ahead with these 2 methods to make them telling all what they can tell, and in order to identify new markers, certainly, in order also to join this method in order to make a modeling for the fertility of the semen of goats and of books. And this modeling will be a great help in order to be more sure that we keep in a collection for the cryobank semen that are much more good than bad. That's all for me, I think. Thank you for your attention.
Okay, thank you, Elisabeth. One question. Beate?
These good or bad ejaculates, were they from different bugs? Or did you have a good and a bad ejaculate of the same bug?
This is a problem at the moment, you know, with the populations that we got. It seems that the variability inside the book, in the first approach with these animals, seems to be higher than the variability between bulls. Only for one of the breed, you know, we had no time yet to work on the second. This is what you...
It's exactly my experience that the variability between ejaculates of the same bug is extremely high in goats. You don't see that in bulls, for example.
No.
In goats it's extremely high.
And so do you have an idea why? Because you know, we have not that in chicken also. The variability, individual variability is usually lower than the variability between males. So this is a question. Probably we will have some indication of answer with these markers, you know, that will be helpful for the future.
Okay, thank you again, Elisabeth. We are running out of time, but I have still one topic to present. So the idea was to be invited colleagues from the UK to present the GenRes Bridge project. And one of the main objectives of the GenRes Bridge project is to develop a European integrated strategy for plant, animal and forest genetic resources at European level. And our colleagues were not able to come, so my name is also there. I received a few slides. I will quickly go through these slides and try to explain to you what it is about and what we also ask from you as interested people from Image and stakeholders in this domain of conservation and sustainable use of animal genetic resources. So, for those who don't know, there are 3 European networks of national coordinators for plants, animals and forest genetic resources. These are networks of national coordinators, and the objectives of the networks are in fact to develop and to implement a more rational system of conservation at European level, to exchange knowledge and information, to collaborate at technical level, and also with a view to share responsibilities and tasks at European level. And this GenRes Bridge project has a limited number of partners, but the animal genetic resources domain, ERFP, is represented by, I believe, 6 partners in the project, and the same is the case for the other 2 networks, forest and plants. So the context of this GenRes Bridge project, it's a coordination and support action. It's not a research project. It came from a call more than 1 year ago. The project started last year. And I want to highlight the bold text in the call text. It is about joining forces between plant, animal and forest networks to develop and implement ambitious approaches and strategies for the management of crop, forest and animal genetic resources. And secondly, to cooperate between the different communities, to foster genetic resources conservation, also within a wider agrobiodiversity context. So it's not just the pillars plant, animal and forest, but also looking at the wider link to the wider agrobiodiversity context. So why is the European Commission asking for this? Well, in fact, there is not a coordinated approach at European level, at EU level, when we speak about agrobiodiversity. There's a lot of, there are a number of DGs in the European Commission that have some responsibility for some aspects of genetic resources. DG Environment, the Convention on Biological Diversity and the Nagoya Protocol, DG Agriculture, DG Sante for seed legislation, but also for animal breeding legislation, and DG Research. And there's a number of areas, policies and strategy areas, that could maybe have some better coordination. And the idea is to develop a stronger agrobiodiversity strategies for plant, animals, and forest at European level. And the IMAGE project is a research project that has almost ended. Is important to feed into this process of generous bridge. So the outcome of IMAGE is important to develop this European strategy for animals, but also the integrated strategies for plant, animal, and forest. So the expectation of the European Commission for this integrated European strategy is that there will be individual domain-specific roadmaps or strategies. There will be more cooperation between the various networks where possible, and to develop an integrated, wider agrobiodiversity strategy. And what's important to realize that we have a new European Green Deal that has been launched recently. The European biodiversity strategy, so not agrobiodiversity, but biodiversity strategy that we have for some years is being revised at the moment and will be concluded in this year also as a new biodiversity strategy. And there's also an EU forestry strategy under development. So there's a lot of things going on at European level that is important also for us as genetic resources networks to link to or to be part of, and to develop an integrated genetic resources strategy under these broader umbrellas. So as the authors of the slides put it, this is the draft aim of the integrated strategy. We still need to have a discussion on this. So the aim of the integrated strategy is to enhance the conservation and sustainable use of plant, animal, and forest genetic resources in Europe through cross-sector integration, where appropriate, to achieve management synergies, resource efficiencies, and effective policy implementation with the common objective of securing genetic diversity for the survival of humankind. So this is the overall draft objective of this integrated strategy. And I'm not going to read this, it's too much text, but there's a lot of sub-objectives or objectives behind this general objective we have to take into account. So the reason why you have this on the agenda here is that what I said earlier, IMAGE results, recommendations should feed into this strategy development. So all the recommendations from IMAGE should be taken on board to be part of the integrated strategy. We already had a large stakeholder workshop in October where various stakeholders from a different domain were present, and also these stakeholders presented already their views, and we had some discussion across domains, what are the main issues in an integrated strategy. And also the ERFP structures, European Regional Focal Point, the network of national coordinators and experts, they will contribute in the next 6 months to this integrated strategy development. So this is the process that is ahead of us for the next 6 months. I thought there was another slide, but the idea is that the integrated strategy will be developed this year. So by the end of this year, we will have a draft integrated strategy ready. And it's up to us as an ERFP network and all stakeholders that are present to feed into this strategy development process. So we heard all the recommendations already of IMAGE. We have presentations, we have discussions, but maybe there are some more general recommendations from you now. We do not have a lot of time, but if you want to put something forward now that should be really part of this integrated strategy from an animal perspective,
you can also raise it now. And many people who are now in the audience are also involved in the further process through ERFP. So thank you, and maybe there are some suggestions and comments here. Any comments, suggestions? Michel.
I think that from the viewpoint of animal gene banks, it should be relatively easy to identify common steps across domains. For the in situ, I see it much more difficult. Maybe we should focus on what we have developed in image on complementarity between ex situ and in situ. What I found interesting in the workshop last October that there are similar objectives, similar wording of objectives, but under the objective people are doing different things. Either because the sociology of the actors is different, or the biology of the species is different. So an integrated strategy would be to agree on the main objectives, but the implementation probably will be different. But we have some lessons to take, at least for gene banks. That's what we see in France, maybe what you see in Netherlands, where you have gene banks for plants, animals, and forests in the same organization. There are some common, reciprocal interest. For example, for the plant gene banks, they are using the resources much more for breeding than what we are doing in animals, where it is for conservation. But in plants, they do not freeze for long term. They have to regenerate every 10 or 15 years, so which is very costly. The advantage is that they check the phenotype, but then they do not conserve more than 10 or 15 generations in animals, because we have the liquid nitrogen. We can conserve for much longer time. But then maybe it's more expensive for animals to take it out and produce animals than for plants. So I think there's this sort of experience that we could share to see whether we can still change or test other methods, really from the viewpoint of gene banks. But I'm very surprised that in forest there is a complete separation between the in situ conservation and the gene banks that are for research, only almost, and the in situ, which is for forestry program and almost no research. At least in France, it's completely different governance. Which, when I discovered that, I was really surprised, even shocked, because I think it is more productive to have more exchanges. But apparently, this is the history of the forestry world. I don't know. That's because the trees, they grow for longer, and so it's completely different perception of one individual tree or one sheep. Of course, it's so. I think it is enriching to discuss, but I don't think we can come to practical recommendations, which are for the three domains. But at least see how we translate a similar objective in different actions.
I think it's very clear that technically the domains are very, very different. So I fully agree. An important framework we also take for our integrated approach is the FAO framework. So it started with plants and animals, forests, and now FAO is also working on an integrated biodiversity for food and agriculture strategy. And if you look at the higher level, we have more in common than we think. And we should be more visible, this agro-biodiversity domain should be more visible in the larger biodiversity context. And I think that is one of the main objectives here, to see how we can have a better position from this area at European level, and to develop some, I think not at a technical level, but at a more strategic level, where are the common issues and synergies and objectives. wells. So, what I said before, ERFP is part of the game. There's a group in ERFP who has to run the show to develop the strategic priorities from ERFP, from the animal side, and we take into account what comes from the projects like IMAGE and from stakeholders. And in the end, yeah, we have to end up with an approved integrated strategy, approved between the 3 domains of plants, animals, and forests. And it sounds like a long process, but we have only 1 year to do this, so it's quite a challenge. So, if there are no further comments here, I want to give the floor to Michel for closing remarks of the whole session of today. You're welcome.
It has been a long day. So I would like mainly to thank the speakers, because I'm quite happy of all the presentation we had, which I think have really shown the different components of the gene bank, what is the life of a gene bank, and what are the missions, and what are the challenges for gene banks. And in the different work packages, I think that objectives have been reached as compared to what we promised. Of course, we have to wait for the Commission and the experts to agree with us, but I think we have very good examples of the different objectives that we set up at the beginning of the project, but we also identify future work needed, both for the fertility, for also the economic analysis, probably economic analysis, which was really probably the first real organized study at the European scale. And we have a lot of questions to enrich these economic studies. But for molecular tools and breeding tools with the SNP chip and the MOPS program and the portal, I think we will have already solid building blocks to help for the future development of gene banks. And so we have a busy month of February, all the partners, and then we will put on the website of IMAGE more information, because at the end of the project, that's where we finally we have a better vision of what we can make publicly available on the website, as compared to the beginning of the project, where sometimes we say, well, we are not yet sure whether we put this or not on the website. Now we can really provide a lot, I think, on the website. And so, if I understand well, Luis considers maintaining the website even after February 29, which is very important, so that people can still go back to this website to find our results and main features or case studies or deliverables. This is important to continue. And there are some other EU projects which I hope will inherit some of the recommendation of IMAGE. And now we have also all of us to to lobby or to send messages back to the EU, so that there is still work to continue for gene banks, and we can also send some maybe precise messages showing what have been done, and I think all the colleagues here have done a lot, and what we would like to continue for a good development of gene banks. So tomorrow for the partners, we will have this real action plan until February 29, all the final version of deliverables and reporting, so the story is not finished for them. And for the, well, if we can get feedback from you or from colleagues, I think it's always possible. We have to organize this maybe on the website to get some contact address. But I would like really to thank all of you and Susanna for the organization, because I think it was probably tiring, but I hope very enriching day for all of you. And so I thank you for staying until the end. Thank you. And so we'll have abstracts. I have asked abstracts to all the speakers. So those who did not send the abstracts to Anthony, please, you send the abstract, because then we will make the abstracts available on the website and also on the deliverable. One page abstract, English. You can put text, picture, but one page abstract. Thank you.